Hello, welcome back to our final video for the entire unit. So you should feel really good about yourself if you've watched all of these. But this is all about the electron transport chain in the mitochondria. So we talked about the electron transport chain for the light-dependent reactions in photosynthesis. We learned a lot about them. It's really cool. And so that actually is going to make this video a whole lot easier. So much easier that I'm really just going to be using this slide to explain everything. So if you're really confused on the idea of an electron transport chain, go back and watch that video, light-dependent reactions, but I'll try my best to explain again here. So hopefully you recall from the Krebs cycle, we stockpiled a lot of these things called electron carriers, NADH and FADH2. We did get a little bit of ATP, we released a lot of CO2, and CO2 doesn't play a role at all anymore because now we've completely oxidized glucose. Glucose is gone, carbons are gone, they're all gone as CO2. What really we've done is we have completely oxidized glucose so that we could reduce molecules, NAD and FAD, so that we could get these electron carriers. These are going to be so important to the electron transport chain. So let's walk through this. So the electron transport chain is going to start with these electron carriers. These electron carriers, as they are named, are going to carry electrons, and they are going to drop off the electrons at the first complex. So we have NADH, it'll drop off its electrons. Well, if it drops off a negative charge, that leaves it with this positive charge, NAD+. Now this electron is going to move down the electron transport chain. Now these don't get excited because they're actually already high energy electrons. So this is a high energy electron, which means that it is getting passed from complex to complex. And every time it gets passed from complex to complex, it is going to be a little bit lower energy. Now let's think about these protein complexes. The first complex here is accepting a high energy electron. And remember, almost anything will accept a high energy electron, so we consider this to have a low reduction potential because it'll accept almost anything. It doesn't really want an electron, but it'll accept it because it's a high energy electron. Now, once it gets to the fourth complex here, well, this fourth complex, it's accepting a low energy electron, and most things don't want a low energy electron. So it has to really want to be reduced. Remember, reduction is accepting an electron, so it has to really want to be reduced because it is a low energy electron that it's accepting, and so we say that this protein complex has a high reduction potential. So it is moving from a complex that is a low reduction potential because it's accepting a high energy electron to something that has a high reduction potential, has a high probability of being reduced because it'll accept a low energy electron. And as that is happening, that produces energy. And so that energy is going to be harvested to pump protons into the intermembrane space. So moving electrons from something that is low reduction potential to high reduction potential is essentially electricity. And so that electricity is being used to pump protons into the intermembrane space. That will become very important when we get to ATP synthase. So just so that we're following along, NADH comes in, it drops off its electron, and now it is returned back to NAD+. That NAD+ can go back to the Krebs cycle and go get reduced again during that process. The second complex will accept FADH2, and it will oxidize FADH2 to FAD, and so that is also going to supply electrons. So both of these electrons will move down the electron transport chain to a final complex that is going to have a very high reduction potential. But that's not where the electrons end up. Instead, the electrons end up at oxygen. So think back all the way to Unit 1. Hopefully you remember that oxygen is super electronegative. Remember, that's why it generates polar bonds in water, because it pulls on the electrons more tightly. It really, really wants electrons. So oxygen is the final electron acceptor. Now, the chemistry here is super weird, and so we're not going to go into it, but you do need to understand that these electrons are passed from protein complex to protein complex, ultimately to oxygen. And this is very important because it'll accept some electrons, it'll pull some protons from solution, and remember we talked before that we just kind of have to believe that there's protons around, and when it accepts two protons and two electrons, then it can form water with half of this oxygen. Now I told you the chemistry is a little weird here because a single oxygen is not very happy. And so we kind of have to think about multiples. So really think of it as 4 protons, 4 electrons, 2 oxygen, and that would make 2 H2O molecules. Now this should be pretty exciting because if you remember in photosynthesis, the light-dependent reactions, we started with water. And water was the one supplying the electrons. Well, now it's full circle. Now we have oxygen accepting electrons to generate water. And so that is pretty neat. And that is how we get water out of photosynthesis. So if you think back all the way to the very first video on cellular respiration, which was talking about that net reaction, we saw glucose was an input, oxygen was an input, and we got CO2 out and water out. And here is where that H2O comes from. Now I told you that this electron or this gradient here, this proton gradient, is going to become very important when we get to ATP synthase. And it is. So let's talk about how that happens. Well, just like in photosynthesis, we have this high concentration of protons in the intermembrane space and a relatively low concentration in the mitochondrial matrix. And so just like in a water wheel, these protons can flow back through ATP synthase. And just like a water wheel, spin ATP synthase. And that mechanical energy can generate ATP from ADP. So we can use that mechanical energy to store energy in that third phosphate, that bond between that third phosphate of ADP to ATP. That is really cool. So hopefully, if you remember for photosynthesis, this is a really, really similar process. Now I want to orient you to the mitochondria because if we don't start paying attention to locations, again, we get really lost really quickly. But when we see the pumping, we are going to pump the protons from the electron transport chain, using that energy from the electron transport chain, into the intermembrane space. So generating a high concentration of protons in the intermembrane space. And then it will flow back through to the mitochondrial matrix. So if we're thinking about where ATP ends up, it ends up in the mitochondrial matrix. Now this whole process is called oxidative phosphorylation. So we talked about substrate level phosphorylation. Now we're talking about oxidative phosphorylation, where cellular process, which is a cellular process that harnesses the reduction of oxygen. Remember, oxygen is the final electron acceptor, so we are reducing oxygen to generate high energy phosphate bonds in the form of adenosine triphosphate. So we use this energy from the reduction of oxygen, which is very energetically favorable, and we use that energy to bond an inorganic phosphate to an ADP to form ATP. So now we have this energetic currency, and the good thing for us is this is super advantageous. How advantageous, you might be asking? Well, I can tell you that each NADH, you get about 2.5 ATP. For each FADH2, you get about 1.5 ATP. So the reason that we were stockpiling so much NADH and FADH2 is so that we could get so much ATP out of this really efficient oxidative phosphorylation. So this becomes very, very important. Now, as you are working through this material, as you're studying, it's very important that we understand both the electron transport chain in mitochondria and chloroplasts. They are very similar. There are lots of principles that we can apply to both, but it is also important that you understand the differences, because that could be potentially a really good FRQ question. It could be something that you see on the AP exam. It could be in a multiple choice question. But it's very important that you are understanding the differences as well as the similarities. I do not suggest just learning one and then hoping that the other is the same, because there are differences. So you can look at these questions that I have here, pause, and then move forward. Now, what I want you to start thinking about is the what ifs, okay? So if we go through the entire electron transport chain, but we potentially uncouple ATP synthesis. Now, that would mean that we don't get ATP out of the whole process anymore, which is why this evolved. We want ATP, but could this potentially be advantageous? Well, I want you to think, okay, well, the energy doesn't just go away. We need that. Energy can't be destroyed. And so it is not going to just go away. Instead, it will be released as heat. That could be advantageous. It's advantageous in babies. You did this, I did this, everybody did this at one point. The babies have a special type of tissue called brown adipose tissue, sometimes shortened to BAT. And these are going to be specialized tissue that uncouple ATP synthesis in the electron transport chain. So instead of having a bunch of ATP synthesized, they release it as heat. And that's because babies don't do a good job shivering. They are not moving. They can't generate heat in the way that adults and other people who can move very easily can. And so this is a type of non-shivering thermogenesis. It's really cool. I recommend spending more time looking at it. But you can also imagine that this maybe isn't always advantageous. There can be some dangers involved with this because it turns out you need ATP to live. And so there is actually a drug called dinitrophenol who thought that this could actually be pretty advantageous. If we make our cells less efficient at making ATP, maybe we can capitalize on that and make a weight loss drug. Because basically you're telling your body that when I burn a glucose, I don't get as much energy out of it, so I need to burn more glucose. And so they thought this was really clever. They thought it was basically a hack to eat as much as you wanted and never really gain weight. Problem is a lot of people died. So this happened in the 1930s. As far as I know, it's about 38 people died. It got taken off the market very quickly after that. It is apparently still available in some very dangerous markets, but I don't recommend it. It's very dangerous. If the dosing is off just a little bit, you die. So there are other ways to manage that. Okay, now you might recall that I said that linkage reaction and the Krebs cycle, even though they don't directly use oxygen, we still consider them aerobic. And the reason why is because if we don't have oxygen, then the electrons can't be accepted and then go away as water. So if we don't have oxygen, then we end up having a backup. Those electrons stay in the electron transport chain. The NADH can't drop off their electrons, and so we quickly use up NAD and FAD, and those aren't going to be able to go back, remember, because NADH drops off an electron, gets converted to NAD, and the NAD is supposed to go back to the Krebs cycle. Well, if NADH can't drop off the electron, now NAD... I just lost my train of thought. Anyway, the NAD cannot go back to the Krebs cycle to get reduced again. And so we end up having this backup that's kind of like a traffic jam. So if we don't have oxygen, then everything else gets halted except glycolysis. Glycolysis can proceed and go through the fermentation route. Everything else will back up. And so we really need oxygen for all of these steps, even though oxygen is not directly involved. Now, we've talked a lot about the oxidation of glucose, but it turns out we can eat other stuff. We don't just eat sugar. We eat fats, we eat proteins, all that stuff. And so you have to understand that they're getting broken down too. And they actually don't really have a different pathway. They just have a different way to get through the pathway. So in the case of triglycerides, well, they have a glycerol backbone and then fatty acids. So these are fats, right? So the glycerol can be directly converted into G3P. G3P is an intermediate of glycolysis, so it can actually enter in part of glycolysis. The fatty acid can be actually directly broken down into acetyl-CoA molecules, and then the acetyl-CoA can go enter the Krebs cycle. We're not reinventing the wheel with all these different substrates, all these different food. And so this graphic I really like because it really shows you all the different ways that we can get through to do oxidative phosphorylation, which is the goal. That's what we really want. Proteins can enter at pyruvate. They can enter as an intermediate in the Krebs cycle. Triglycerides can enter in different parts as well. You don't need to know where it enters, but I do want you to understand that we're not reinventing the wheel every single time. Instead, we're just using the same wheel to do slightly different things. Okay, so now let's talk a little bit about the regulation of ATP. Now, we do not need to be constantly breaking down ATP. In fact, if we don't use it pretty quickly, we waste it, and that's not the goal. Instead, we are going to give our cells signals. We're going to tell our cells when to stop making ATP. And part of that is coming from ATP acting as an inhibitor. So ATP can go back and inhibit earlier steps. So it can go and inhibit an enzyme important to glycolysis, or it can inhibit an enzyme important to pyruvate oxidation or citric acid cycle. It can come back and stop it at any point so that we don't go through and waste energy in the oxidative phosphorylation steps. And so I really like this image. What you can see here is that ATP can actually come in and inhibit an enzyme in glycolysis. However, what you can also see here is AMP will activate it. And so AMP is the monophosphate. You're only going to get a monophosphate when you are really low on energy. That's when you've completely hydrolyzed ATP, not only to ADP, but also one more to monophosphate, adenosine monophosphate. So this is when you're really low on ATP, and so it's going to activate the pathways so that you can get more ATP again. And you can spend some time looking at all of these and trying to make sure that you understand. But the idea here is that ATP can inhibit, and then when we have the alternative like ADP or AMP, that can actually activate the pathway. And so we'll end with this. I actually really like this slide because every time that I teach biology, I always hear you guys say the mitochondria is the powerhouse of the cell. And it is, but now we understand how. And that's because it does ATP synthesis through oxidative phosphorylation. And that's not a horrible day. That's an awesome day. I hope your studying is going well. I hope you have a great time learning all of this, and I'll see you soon. See ya.